And I know I am the last person between here and lunch. So, and we have covered some of these materials. So hopefully I'll click through the slides that are with you. Basically wanted to start with the case presentation. This is, I'm not going to read it all out, but basically in front of me was this healthy, young, dynamic individual who wanted to be an aspiring actor. This was his brain, very similar to the one showed you before. All of these punctate little lesions. And to me, this was my aha moment in the field. Like, what the heck? This guy is my son's age, and his brain looks like a 70-year-old's. And this was shown before, showing the risk of silent cerebral infarction as a function of age. As an engineer, I do not like straight lines that, you know, where's the asymptote to that for, these are half my age. So that's very concerning. Obviously, with white matter disease, there's not really one type of white matter lesion. There are multiple types. You have these larger watershed artifacts, which are thought to be acute supply-demand mismatches. You have these punctate lesions or white matter hyperintensities that people argue about what their origins are. We still think supply-demand plays a role in that. And then as you get older, you start to get this periventricular leukoarosis, which is probably yet still a different mechanism. And I was very curious on the mechanical shear related forces and the roles that they might be playing in that particular form of white matter disease. And we can learn a lot by looking at other diseases that also are enriched for white matter hyperintensities. Here's just a bunch that can occur in you or me. And what do they have in common? Hypoxia in all of these, abnormal mechanical forces on the blood vessels, and a component of vascular inflammation. That's sort of the evil triad that holds up this burden of white matter disease. And of course, in sickle cell disease, all three of the legs of this stool are present. So that's where I started, and I went to the Fick equation originally, supply, demand, let's go after these things, focusing first on delivery. These are the equations which you are probably all very familiar. Oxygen delivery is the content of the blood times the cerebral blood flow. So as content goes down when you become anemic, you better have a compensatory increase in cerebral blood flow or your oxygen delivery is going to go down. So what happens if you look at all-cause anemias? This is our paper, but there's actually several other manuscripts out there that show that there is a reciprocal relationship between cerebral blood flow and your oxygen content. And agreement among the studies is really quite good, independent of the underlying cause of the anemia. And you can actually explain away 80% of the variability by things like your oxygen content in your age. There is a maturation component so that young brains use more oxygen than old brains because there's more gray matter. Despite this, when we look at sickle cell disease and we do tensor-based morphometry or atlas-based measurements, there is a systematic loss in white matter volume. And gray matter is preserved, but the white matter structures are smaller. And this just shows the cortical regions superimposed over the white matter lesions that are lost. And you see an interesting pattern that you can describe by either an arterial explanation, which is the anterior and middle cerebral arteries seem to be more vulnerable than the posterior circulation. Or similarly, if you look at an atlas of the brain and say, old brain, new brain, and it looks like the old brain is relatively preserved. And that kind of makes some sense phylogenetically. To run away from the saber-toothed tiger, you don't need your frontal lobes. You need your brainstem. You need primary, very primitive cortices to survive in this world. The rest is sort of fluff. To refine this a little more deeply, what we did is tensor-based morphometry. You morph the brain to an atlas, and then you look at that deformation matrix and you do linear correlations with hemoglobin across that tensor. And you can say, all right, what parts of the brain systematically expand or compress the more anemic you are? And no parts of your brain expand, basically, but some parts of the brain are definitely systematically smaller when you're anemic. And these turn out to be the deep white matter structures shown here. This was all-cause anemia. Again, we looked at both thalassemia and sickle cell disease, two congenital malformations in the synthesis of hemoglobin. And interestingly, this deformation index is the tensor, the determinant of the tensor matrix, with a low value being shrinkage. And so as you go to a lower hemoglobin, you get shrinkage. And both males and females show this relationship. But what was interesting to us was then we looked at the deformation index as a function of, or we looked at a full-scale IQ as a function of this deformation matrix. What we saw is that there was a linear relationship in males. So the males that had shrinkage had much lower full-scale IQ. The females didn't. So there's something protective about being a woman that disrupts this relationship between IQ and white matter shrinkage. We've obviously, morphometry is very primitive. We're looking at diffusion types of imaging in the brain. You're all fairly familiar with the general principles. This illustrating the concept of fractional anisotropy or preferred water movement along axons. We've gone to slightly higher models than the simple Gaussian statistics and are looking at mean kurtosis. And this shows the statistically significant regions of the brain with abnormal kurtosis in sickle cell disease compared to controls. We controlled for age and we controlled for sex. And you go, wow, that's the entire white matter. There is no localization in deep structures, unlike the volume loss. So this is not a hypoxia-driven change. It can't be, because regional oxygen saturation is very different in the white matter than in the cortical, the sulci white matter. So this is describing really a demyelination, a systematic myelination process, rather than localized tissue hypoxia and white matter damage from that. And what's interesting is if you do the exact same thing in thalassemia, where there's no sickling process, you see the same thing, the same extent, the same severity, the same proportionality to hemoglobin. And with the thalassemia patients, if we control for hemoglobin, there is no residual abnormality in mean kurtosis. So it can be all explained by the hemoglobin level itself. The other thing that's interesting is it was correlated with processing speed. So the more abnormal the kurtosis, the more abnormal the processing speed, which makes sense for a diffuse demyelinating or dysmyelinating process. When you do the same control for hemoglobin in sickle cell disease, you eliminate most of the statistical significance, but there are still regions of the brain that remain abnormal, where there's abnormal kurtosis. When we put another parameter in the model, one that reflects the degree of red cell destruction, such as LDH or reticulocyte count, these are surrogates for what we call hemolysis, the intravascular destruction of red blood cells. All of those pink areas go away. So this suggests that it is worse to be to have sickle cell anemia rather than a thalassemia type of anemia, which we've always posited, but we've never been able to show. And so this is, I think, a very interesting finding. And why is intercellular red cell destruction bad? Well, it dumps a whole bunch of toxic things into the vascular system. Hemoglobin and heme both attack the vascular endothelium. They catabolize nitric oxide. And so there's a whole lot of reasons why intravascular hemolysis should be worse than just having anemia. So to sort of take home message here is our global oxygen delivery to the brain is normal. There's a reciprocal increase in flow that preserves oxygen delivery to the brain. Despite this, we have evidence both of volume loss in the deep structures, sparing of the white matter, and a weird pan-white matter demyelinating process. And the posterior circulation of the brain, or the old brain, seems to be relatively preserved. And the consequences, the functional consequences, appear to be sexually dimorphic. And this has been shown in sickle cell disease. Males definitely have worse neurocognitive outcomes than females. All right, so what is the pathophysiology? How can we have normal delivery and downstream damage? Well, one possibility is that the cerebral vascular reserve is decreased. And a second possibility is that the whole brain oxygen delivery is preserved, but maybe not equally. Maybe the gray matter is relatively protected, or there's even a steal phenomenon between the gray and the white matter. The other possibility is that there's selective destruction to the microvasculature in the deep structures compared to the superficial structures, perhaps because of mechanical forces or other things. The other possibility is that there's a problem not with the delivery, but with the extraction of blood. And lastly, there could be just the consequence of a million heartbeats on pounding the brain and the transmission of these mechanical forces to the distal vasculature, which we know is a big component of normal aging, and maybe that process is accelerated in sickle cell disease. The stroke volume on a beat-to-beat basis is 50% larger than in you or I. So mechanically, that's more blood going in, more coming out. Perhaps that is contributing to this white matter disease. So starting first with the cerebral vascular reserve, obviously this can be done with a fancy device like the Respiract, where you use a controlled dose of carbon dioxide, or it can be done by giving a drug like Diamox, acetazolamide, which tricks the brain into thinking there's changes in that it's acidic, and so that it will vasodilate. And the black curve on the left shows the rise in cerebral blood flow by arterial spin labeling in control subjects. The red indicates the corresponding response in sickle cell disease patients. This was data by Lena here in row 3. So, and you can see that there's a relatively much larger change in patients, the control subjects, compared to sickle cell disease patients, indicating a much larger cerebral vascular reserve. Interestingly, there is a sex difference, and women with sickle cell disease have a better cerebral vascular reserve than men. The other thing we looked at was the consequences of one of the therapies, which is hydroxyurea, that increases the so-called hemoglobin F, and the oxygen affinity is higher with hemoglobin F. So in the end, you have to deliver more blood at baseline to give the tissues enough oxygen. And so what we find is that in these subjects, the baseline who are treated, who have a higher hemoglobin F fraction, you can see that their resting oxygen delivery is increased because the brain blood flow goes up. But then their ceiling, which is the post-Diamox oxygen delivery, is not improved. So cerebral vascular reserve actually goes down in people treated with hydroxyurea, but their resting delivery is higher. So what about this idea of the global versus the regional delivery? This was a study just looking again at arterial spin labeling. This is a T-map, which it reflects compared to controls systematically. You can see a lot of red and orange indicating that sickle cell disease patients have high cerebral blood flow. No surprise, they're really anemic. Well, if you take that same map and scale it by their oxygen saturation and their hemoglobin, you get an oxygen delivery map, a T-map. And then when you look at the map in the lower right, you can see lots of green and yellow. So normal to slightly increased flow, oxygen delivery to the gray matter. But in the deep white matter structures, you see a lot of deep blue. So the oxygen delivery to the deep structures appears to be decreased. Now, a caveat, a limitation to this study is this was a single delay P-Castle sequence. There's a possibility that this could be a transit time. There could be the influence of the longer transit time to the white matter, and we're exploring that now with multi-delay P-Castle. What about this idea that there might be microvascular damage? Well, this is a study where we took sickle cell disease patients and we gave them a brief bolus of nitrogen, 5 breaths, and you create a very short, sharp drop in the oxygen saturation. It's basically imperceptible. I have done this experiment probably myself close to 50 times. And I can tell you, I can learn that the bolus is passing, but it's really a mild stimulus. And what it does is create a very sharp contrast bolus that we can characterize the time to peak and the time to recovery. And this is what it looks like in patients who have no silent cerebral infarctions on the top versus ones with silent cerebral infarctions. And in the ones who are SCI negative, you see relatively homogeneous changes. There's some differentiation between gray and white matter. These are T maps. So the blue in the top center panel, that indicates a shorter time, half time of recovery. And that's because of the higher cerebral blood flow in these patients. But honestly, you don't see, when you look in the SCI positives, you see some remarkable differences between the anterior and posterior circulation, and saying that the kinetics of this process are very different in those who are prone to white matter strokes and those who aren't. You even see left-right differences in this cohort. And just in general, it's looks more like birdshot hit the brain. And I think that this is evidence that those who are having silent cerebral infarcts have more microvascular damage. It's not proof. It's really just a quick look at that and a hint that maybe the passage of bolus through this brain is much more heterogeneous in these patients who are prone to silent strokes. What about this concept of extraction reserve? This is focusing on the downstream side of things. What's the color of the blood coming back from the brain? Same oxygen delivery principles. We need to now know the saturation of, and we use the sagittal sinus as a surrogate for whole brain measurement. We have used the trust measurement, but this can also be done with susceptibility-based imaging. The key with trust is that you're measuring T2, and you have to convert it using a known hematocrit into an oxygen saturation. You pass that into the big back calibration box. And this is where the controversy in the field is coming from. The first calibration came from bovine blood, and I believe the bovine blood gives pretty reasonable results for oxygen for hematocrit levels between 35 and 55, which was where the calibration was derived. But there have been some papers which have tried to extrapolate that to sickle cell disease, which is well outside the calibration range. And that has systematic error, even in vitro studies where you can directly measure the oxygen saturation in a benchtop oximeter. The hemoglobin A model was derived from normal controls. It's over a much broader range, and it's been independently validated. The controversy comes with hemoglobin S, which was derived from patients in 2 independent studies. The problem is it's a really hard in vitro study to do, and there's some concern about whether it represents what really is going on in vitro. And why does this matter? Because they predict different results. This is the oxygen extraction fraction predicted in patients with severe sickle cell disease, mild sickle cell disease, thalassemia, and controls. And interestingly, the mild sickle cell disease patients who do not have different oxygen extraction fractions than controls, but their hemoglobin levels are on the order of 10.5 to 11 grams per deciliter, so they're not that anemic. In the thalassemia and severe sickle cell disease patients, the mean hemoglobins were on the order of 8.5 and 9. And so with the more severely anemic patients, we know the cerebral blood flow goes up, but the predicted oxygen extraction fraction is much, much lower. And this could be due to intravascular shunting, which was one of our leading hypotheses. And the interesting thing is that this is not just a brain phenomenon. If I stick a needle into the antecubital vein and then put it on a benchtop co-oximeter, I see the same phenomenon. This is the oxygen saturation in the vein. The nurses came to me and said, I thought I hit the artery because it was so bright red. You'll see venous oxygen saturation, 95% coming out of a needle in the antecubital vein. So this is a biology, this is a physiology, and that's why I was so curious on the previous presentation where we're seeing suggestion of blood just blowing through the capillary bed with a very short transit time and making the veins, it's called arteriolarization of the vascular bed. There's another big confounder here, and you'll see studies that refer to OEF as being a marker of vascular stress. And that's true in carotid artery stenosis. When you have a restriction of blood flow to the brain, the OEF directly is a risk marker that you can take to the bank and say, you know, higher OEF, more risk of stroke. That is not true in sickle cell or any of these congenital anemias. Why? Because the hemoglobin dissociation curve starts to become really, really important, and it's not normal in these anemias. And for those of you who don't remember, that's a plot of oxygen saturation versus oxygen tension in the blood. And depending on the location, it's a sigmoidal curve. We describe it by a P50, which is the PO2 at which the oxygen, the oxygen in the blood is 50% saturated. And as it moves rightward, the blood will unload oxygen more easily. As it moves leftward, it holds onto oxygen much more tightly. So fetal hemoglobin is way over on the left. So is transfused blood. Transfused blood is very high affinity because it's low in 2,3-DPG. Sickle hemoglobin shifts way to the right. So whenever you're doing a blood transfusion in somebody and you're trading hemoglobin S for blood that's been stored, that's going to move the hemoglobin dissociation curve. And it matters because the brain doesn't care about oxygen saturation. It cares about PO2. It wants to operate at a very characteristic PO2. And the oxygen extraction fraction will just follow that difference. And this is a plot of OEF by trust in our patient population. The green is controls. What I plotted on the right is the left shifted hemoglobins. This is hemoglobin F, but also because we had some patients with other high affinity hemoglobins, I lumped those all together. They actually have similar oxygen affinity as hemoglobin F. And I think you can appreciate is that left shifted hemoglobin fraction goes up, our OEF crashes down to as low as, in the 20s. So you have to control for this effect. I think there is a shunting effect, but you have to control for the position of the hemoglobin dissociation curve as well. And this is just a schematic. I think we saw something earlier today with this idea that with higher blood flows, you don't just go homogeneously through the capillary bed and unload your oxygen in a leisurely manner. The brain's in a can. If you try and ram a lot of blood through all of those little pathways, you're going to get filtration. Filtration is going to build up pressure, and that's going to push your brainstem out of the can. So you can't do that. And so I think what happens is that high cerebral blood flow, these bypass channels become more important. And it's not true vascular shunt, as was questioned earlier. It's a physiologic shunt. It's just a transit time phenomenon. And I think that we see greater fraction of blood with short transit times in patients who have higher cerebral blood flow. So that's what's causing the purple blood in the veins. And this is a graph that I'm actually fairly proud of. It was a lot of work. So this is the cerebral metabolic rate in all of the patients that we'd studied with these crazy different heterogeneous anemias. And you have to correct for age because CMR2 does change with age. But the green dots are our data. The circles are data pulled from the literature from every study I could find that had either a PET measurement of CMR02 or a Keedy-Schmidt, which is a dilution method with direct physical validation. So very highly accurate reference data. So, and this is using this controversial sickle calibration. And so I think you can appreciate we're replicating what other people have seen with gold standard methods. So what have the chronic anemias taught us? I'm not going to pound this out in great detail, except that, you know, there is shrinkage, there's dysmyelination, women have lower functional. There seems to be a decrease in extraction reserve at high flow. I want to turn real quickly to what about acquired anemias? And they're really common. So 5% of the population is anemic. There are multiple causes: iron deficiency, folate deficiency, anemia chronic disease, and bone marrow failures. But I'm most interested in people who have moderate to severe anemia, which is a hemoglobin less than 11. And in particular, I'm interested in the younger age group, because this is otherwise healthy people running around with a significant anemia, and they're not going to the doctor, and they may not feel bad. So what are the health consequences of that? And in this age group, this is almost exclusively women and iron deficiency anemia. So why is this relevant? Well, for those rates I showed you, minorities have a rate that's 3 to 5 times higher. And we know in children, I'm a pediatrician by training, and in children, iron deficiency causes permanent, irreversible structural damage and functional damage to the brain. So when I submitted my grant on this, the reviewers said it's easily, in adults, it's easily detected, it's easily treated, and it has no functional consequence. And I looked at that and I said, there is nowhere in the literature that suggests that any one of those is true. And if you've ever tried to treat iron deficiency anemia, it's horrible. Oral iron sucks. And women take it for a little while and they stop taking it because it does nasty things to them. And do we know that it reverses? It's partially reversible. That evidence is there, but complete reversibility has never been described. So this is a pilot study we did in 37 adult women with IDA. And let me just get to the money here. So otherwise healthy, iron deficiency phenotype, broad range of hemoglobins because of our screening criteria. And we did look at white matter hyperintensities. In the younger age groups, there was no increase in risk, but the older women tended to have apparently a increased risk of, and when we looked at cognitive function, this was the most fascinating. There was a real breakpoint at 10 grams per deciliter. For all you ladies who may have been mildly anemic in your lifetime, as long as you were above 10 grams per deciliter, there really was no perceptible cognitive impairment. But when it drops below 10, it's a huge effect. That's a Cohen's d of 1. So 1 standard deviation below predicted. That's the difference between an A, a B, and a C. And you know, if it's my daughter, I care about a standard deviation of cognitive impairment across a very broad battery of performance. And furthermore, there were structural changes in the brain in adults, including a loss of volume in the temporal lobe. And that's those are the cognitive tests that correlated with that. There were also white matter changes in the cingulate, the corpus callosum, and the cerebellum, all of which had cognitive correlates. Almost done here. So one really fascinating thing, we looked at the cerebral blood flow. I just showed you all of this data on chronic anemias and how the brain blood flow goes up. Well, guess what? It really doesn't go up very much in women with iron deficiency. Here's those historical controls that I showed you. Here's the increase in cerebral blood flow in this iron deficiency cohort. And there's a very perceptible increase in cerebral blood flow, but not to the level that you would expect in hemoglobinopathy. When you look at cerebral metabolic rate, though, it falls perfectly on the previously showed confidence intervals. So unlike in sickle cell disease, you have increased flow but decreased extraction. In iron deficiency, you really have kind of normalish flow and normalish flow, decreased oxygen delivery, but normal extraction. So you end up in the same place in your cerebral metabolic rate, but for two different reasons. So what do I think is going on? I think that we do, to sense hypoxia, the hypoxia sensor sits on the arteriolar, kind of upstream of the vascular bed, and it requires a heme protein. So if you're severely iron deficient, I don't think you make enough of those heme proteins to sense the relative hypoxia being anemic. And so you don't have the same compensatory increase in cerebral blood flow. But because you have a very redundant capillary network, you can actually extract okay downstream. So almost time for lunch. So how do we uncouple this hypothesis? We have a problem because the anemia and the iron deficiency are so tightly correlated that we can't tell what's the problem of being anemic and what's the problem of being iron deficient. So we're going to use IV iron and split those two, because like 48 hours after getting a dose of IV iron, you've corrected the iron deficiency, but you haven't corrected the anemia. That takes a while to crank out all those red blood cells. So we're going to create a nice observation window where the iron deficiency is corrected, but the anemia isn't. And we're going forward with this study right now. So really, we want to to better characterize this scope of neurocognitive deficits, to look at the changes in perfusion, reactivity, connectivity, and permeability in these and relate them, and then see the reversibility with both IV and oral iron and compare the difference. So I think I'll stop because it's lunch.